Hey everybody, I'm hoping to clear up some glycolysis confusion for you. So here is an overview of glycolysis. Before that, let's look at the overview of cellular respiration just so we know where glycolysis fits in this. So up here, this blue box right here is showing what you get out of glycolysis. You're going to split a molecule of glucose. You end up with two pyruvate molecules and some reduced coenzymes. From there, you can enter the citric acid cycle if oxygen is available, then the electron transport chain. So these are three of the big cellular respiration processes. And right now we're going to focus on this first one, glycolysis. Glycolysis can take place whether or not you have oxygen available, so you can continue to produce even just a few ATP even if no oxygen is available. Glycolysis is made of 10 reactions, and in these reactions you are going to lyse glucose. You're going to split it right in half. You're going to end up consuming 2 ATP, but you generate 4, so you end up with a net 2 ATP when you come out of it. And then pyruvic acid can go down one of two pathways depending on whether you have oxygen or not. We'll talk about that in a minute. Let's look at these reactions. Now you don't need to know all of the specific names of the molecules for this class, but I do want you to know the overall steps that take place. So here, phosphorylation, priming, cleavage, oxidation, and dephosphorylation. And I'll explain why you get these names. All right, so first, you have a glucose molecule. Each of these gray circles symbolizes a carbon atom. Glucose is a 6-carbon molecule, so here are your 6 carbons. The first thing you're going to do is phosphorylate it. And so you are going to take a phosphate off of ATP right here. And this phosphate is now on glucose 6-phosphate. Named that way because on the 6th carbon you now have the phosphate, which you got from ATP. So that's why you see a DP here now. So first step is to make glucose 6-phosphate. Next step, you're going to rearrange this sugar a little bit, and then you are going to add another phosphate group. So here's your rearrangement of the sugar. Here is your second phosphate. And again, you're going to get that from ATP. So here's that second ATP that you need to input into glycolysis. You can see that results in an ADP molecule, and now you have the other end of this sugar, which has been phosphorylated. So you have a phosphate here in the yellow circle, phosphate on the other side, there's the yellow circle. And now this sugar molecule has been primed. So it is ready for the next step. So we've added 2 phosphate groups, rearranged the molecule a little bit. Now this we're going to cleave. So we're going to split this glucose in half. So this is where the lysis of glycolysis takes place. And you end up with 2 3-carbon sugar molecules. So we had 1 6-carbon sugar, now we have 2 3-carbon sugars. This makes phosphoglyceraldehyde. And you can see that each one of these sugar molecules has a phosphate group on it right here. So there's cleavage. Moving on, we just talked about this step up here. We have 2 phosphoglyceraldehyde right there. Our sugar's been cleaved. You're going to add 2 more phosphates, and notice this 2 molecule right here. They didn't repeat the steps. They didn't have each one of these phosphoglyceraldehydes. They don't have them separately illustrated, so just remember that there are 2. Each one of these sugar molecules now has a phosphate on either end due to the addition of this phosphate group right here. Now this is the oxidation step. Remember that there are many redox reactions that take place during cellular respiration. Oxidation is losing electrons. Reduction is gaining electrons. Remember your oil rig. So this sugar has been oxidized, and those electrons which have been lost, these are going to go to NAD. So here is the oxidized form as it adds these electrons. Now you have NADH plus 2H. And remember, you're getting 2 because we had 2 3-carbon sugar molecules. So each one of these will be oxidized. Those electrons are going to go to form these reduced coenzymes. So here's your oxidized sugar, and here is your reduced coenzyme. Remember, you're always going to have an oxidized molecule and a reduced molecule. You can't have one without the other. So here's your reduced coenzyme, and we have our oxidized sugar right here. From here, we're going to start removing those phosphates. So the next step is dephosphorylation. We're going to take these phosphate groups, these yellow circles, we're going to remove them from the sugar. We're going to remove the one from each side here, and remember there's 2 sugar molecules, so we're going to end up removing a total of 4 phosphate groups. And so you do that on both sugars on one side, and you put those phosphates back onto ADP, which will form ATP again. So here's your 2. Take the 2 off of the other side of each molecule, and that is going to form another 2 ATP. So remember, when we initially phosphorylated the sugar, we had to input 2 ATP, but now we have gotten back 4 ATP, generating a net of 2 ATP overall. We're 2 ATP more than what we started out with. And we end up with 2 pyruvic acid. Now, the fate of pyruvic acid depends on whether or not oxygen is available in the cell. So far, all of these reactions can take place if oxygen is present or not. It is not dependent on the presence of oxygen. If there is oxygen present, then pyruvic acid will head down this pathway. We're going to head toward the mitochondria. So far, all of these reactions in glycolysis have taken place in the cytosol. But if there is oxygen present, these 2 pyruvic acids are going to be headed toward the mitochondria. And we'll talk about the citric acid cycle in a few minutes. If no oxygen is available, then we're actually going to take back those electrons that we originally donated way back up here with the oxidation step. So these 2 electrons are going to be added back onto pyruvic acid, and when you do that, it makes lactic acid. Lactic acid is then going to head to the liver, and it's going to hang out there until oxygen becomes available. Then it can be converted back to pyruvic acid, and then you can head toward the mitochondria then and continue with cellular respiration. And then you can see as those 2 lactic acid, if oxygen becomes available, those 2 electrons will head back onto this coenzyme right here, so they can be reduced back again. So no oxygen, pyruvic acid gets converted into lactic acid. If you do have oxygen present, then each of those pyruvic acid can both head to the mitochondria to participate in aerobic cellular respiration. So overall, there's glycolysis. First step, phosphorylation. Add a phosphate group on one side, rearrange the sugar a little bit, add a phosphate on the other side of that 6-carbon sugar. So you phosphorylate and prime the molecule. And for those phosphates, remember, come from 2 molecules of ATP. You cleave the sugar, split it in half, add another couple of phosphates. Now you have phosphates on either side of both of those sugar molecules. Oxidation occurs then. You're going to oxidize this sugar and make a 2 reduced coenzymes, because we had cleavage up here, right? You have 2 sugar molecules. Each of them will be oxidized, making 2 reduced coenzymes. Then you take the phosphates back off from each side of each sugar. So there's 2 ATP, and then from the other side here is 2 more ATP. And finally you end up with 2 pyruvic acid after you've dephosphorylated those. So those are the steps in glycolysis. I hope this helped.